All right. Well, thank you so much. And thanks to Arjun and Shalev for organizing this great meeting and for inviting me today. Again, it's a great honor to be here. And the work I'm going to talk to you about today was spearheaded in my lab by Megan Turner and Nick Lammers, but it's also the result of a great collaboration with Chris Wiggins at Columbia and with my colleagues Mike Eisen and Xavier Darczak. And the overall question that we're interested in is how do you go from a single cell to a multicellular organism? And we asked this question in the context of the fruit fly. You can see here how the nuclei in the fruit fly are going through multiple rounds of synchronized division. And at this point, all the cells look the same to you, yet each one of them knows what they're going to be when they grow up. And this is so because they are expressing a unique combination of transcription factors. And these decisions then are manifested by these massive rearrangements, for example, called gastrulation in this case. Now, the cool thing is that we know the identity of the transcription factors that endow cells with their identities. Not only that, we know that they're organized in different layers with patterns with increasing sharpness. There is a first layer of activators and repressors expressing shallow gradients that leads to genes expressing broad domains that gives you genes in 7 stripes, that gives you genes in 14 stripes that ultimately segment the embryo and think of the vertebrae of your vertebrae as your segment and endow the segments with an identity. Are you going to be a wing segment or a leg segment, for example? We don't only know the identity of these transcription factors, we also know the regulatory connections. So this is an amazing state of the art to start asking the types of questions that we're interested in. Yet I have a complaint. And that complaint is that just like this is not a pipe, this is not a fly. And what I mean by that is if I change the names in this network and I give it to the world's experts on flies, they would have a hard time telling me that this is going to give you a fly. You cannot calculate a fly out of this network description. And what I'm going to try to convince you of today is that the reason you cannot do that is because we're missing information about space, time, and concentration in these network descriptions. So our objective is to endow each one of these arrows in these networks with molecular and quantitative information that allows us to reach a predictive understanding of developmental decision making. Basically, can we predict developmental phenotypes from the network topology? And so to be more specific, what we're after is understanding or predicting how the DNA sequence gives rise to the functional relationship between input transcription factor patterns and output gene expression. Okay. And now you might say, well, this is crazy. Like you're never going to be able to reach this sort of understanding just because biology is too complicated. And I would say that's fine. You know, that's a challenge. But all I can say is that the one time that we tried to do this in bacteria, it worked. Okay. So here, this is an example unrelated to flies. This is from bacteria. This is the lycopron where I'm going to show you some work from my PhD where we looked at how like repressor represses polymerase or doesn't allow polymerase to bind to the DNA. And in this context, you can think of different regulatory knobs that you could tune to test your model, such as the binding energy of repressor to the DNA, the number of repressors, and the number of promoters. And you can imagine it's not the same to have 100 repressors and 1 promoter than to have 1 repressor and 100 promoters. Here, I'm showing you the gene expression as a function of the number of repressors, as predicted by our theory, as you tune these different regulatory knobs. The point here is that these are 0-parameter fits. There are no free parameters here. Then we went and took the data, and to a large extent, we showed that we could predict this output. Not only that, all these curves look like different adventures in and of themselves. But it turns out that that's because we hadn't found the natural variable of the system. If you plot this data against the fugacity of the repressor, which is something equivalent to mapping a non-ideal gas onto an ideal gas, what you show is that all the data collapses onto the same master curve. Okay. So again, the point is that this took 10 to 15 years to get to that point, but at least in this particular example, this sort of predictive understanding that I'm advocating for is doable. And of course, you can ask, well, you know, this is for bacteria, eukaryotes are a lot more complicated. How are you going to deal with that? And I agree. There have more bells and whistles, maybe. But the question is which bells and whistles do I need to account for in order to reach this sort of predictive understanding? So for example, in this simple view of an input-output function, maybe what you want to know is not only the concentration of the transcription factor, but you want to know how many transcription factors are bound to the DNA. Maybe you don't only need to know what the pattern of gene expression looks at the macroscopic level, you also need to figure out how individual cells are expressing RNA in order to generate these patterns. So the talk today is going to be divided in 3 short parts. And it's going to also be very much inspired by this great article by Joel Cohen called Mathematics is Biology's Next Microscope, Only Better. Biology is Mathematics Next Physics, Only Better. So in my lab, we're very curious about how we can use thinking to see things that you couldn't even see with the fancy super resolution microscope. So I'm going to introduce you to experimental, to theoretical, and computational microscopes that allow us, for example, to uncover that the expression dynamics underlying these patterns is characterized by transcriptional bursting. We're going to then use theoretical models to try to predict how the binding of transcription factors regulates this bursting. And finally, I'm going to introduce you to some very new technology that we're trying to develop in order to actually correlate experimentally the binding of transcription factors and the resulting transcriptional output dynamics. And the case study we're going to focus on for most of the talk is probably the most understood pattern of gene expression in all of development, which is stripe 2 of the even skip gene. The stripe is controlled by 4 transcription factors, 2 activators and 2 repressors. Initially, only the activators are present and they give you a broad domain of activation. But later on, the repressors come on and they restrict the domain of activation into this little stripe of about 6% of the embryo length. Now, the thing to keep in mind here is that all the data we had so far came from pictures like the one I'm showing you here, which are in situ hybridization measurements, which measure the amount of accumulated RNA in the cytoplasm. Okay, so the first question that you might want to ask is, how do single cells regulate their transcriptional dynamics to result in this macroscopic pattern of gene expression? And to do that, you need to be able to look at transcriptional dynamics in single cells in a living embryo. And to do this, we use some technology that we developed in my postdoc lab with Thomas Greger at Princeton, and that was also developed in parallel in Natalie Dostatny's lab at the Curie Institute, using technology invented by Rob Singer. And the idea is the following. We're going to add a little sequence called MS2 to the gene of interest. When that sequence is transcribed, it's going to form a little loop that is recognized by a binding protein fused to GFP. The result is that in every nucleus in your embryo, you're going to get a little spot of fluorescence, and the fluorescence of that spot is going to be proportional to how many polymerase molecules are actively transcribed. This is what you get when you put it all together. You see in red the DNA and in green the RNA signal. Now you see this broad domain of activation that I was telling you about, and as development progresses, you're going to see how the domain gets narrower and narrower. The other thing to notice as you look at this movie is that you're staring at an in vivo single molecule experiment, because these spots are reporting on the decisions made by an individual promoter in a living multicellular organism, which means that you can zoom into one of these nuclei and count the number of polymerase molecules transcribing that particular locus as a function of time. And what these measurements reveal are these punctuated dynamics that look like trapezoids. Okay, so what do these trapezoids tell us? Well, you need to remember that this technology is measuring the number of polymerases on the gene. Presumably, what the transcription factors are doing is regulating the rate at which you load polymerases onto the gene. These two quantities are related, but they're not the same. And you can relate them through a very simple model where you say that initially your promoter is in the off state, then you turn it on, and you start loading polymerases one by one, and your fluorescence increases. If the first polymerase makes it all the way to the end and falls off, but you still load polymerases from the other side, now you're going to have a constant fluorescence level. And finally, once you turn off the promoter, you're going to lose polymerases and fluorescence at the same rate that you loaded them. What I'm trying to tell you here is that this data is consistent with our model where the promoter was on for a small period of time, these so-called transcriptional bursts. And, people have seen them, as you know very well. People have seen them all over the place in bacteria and yeast and mammalian cells. They seem to be the rule rather than the exception in biology. And the simplest theoretical model onto which you can project this sort of data is the so-called two-state model, where what we're going to posit is that the promoter can exist in 2 states on and off and can switch stochastically between those states. And only when in the on state, it can produce RNA at a given rate. So if you can measure the rate of polymerase loading, you're going to see that you could measure the burst length, burst frequency, and burst amplitude that is determined by the model parameters. So given that we know that these cells are bursting, how do they regulate this bursting dynamics in order to realize this average pattern of gene expression in the cytoplasm? If you go through the math, what you get is that this mean rate of transcription is given by a term that tells you talks about the fraction of time spent in the on state that is multiplied by the transcription rate when in the on state. What this tells you is that you can realize a stripe of gene expression by upregulating K on in the middle of the embryo, or the stripe, downregulating K off, or upregulating R, or any combination thereof. So the question then becomes, what is the strategy at play in stripe formation? And to answer that question, basically you need to measure K on, K off, and R. Now, this would be trivial if I can measure the promoter state, which is measure per size, frequency, to amplitude, and I'm done. The problem is that what's accessible to me is the number of polymerase molecules on the gene. And you need to do some sort of inference. And this is easier said than done. And maybe I'm slow, but we spent like 8 years trying to figure this out. And only once we started working with Chris Wiggins did we figure out how to do this inference. So for 8 years, we were sitting on terabytes and terabytes of data that we didn't know how to analyze. But now we can do it. And let me show you how this works. This is basically, I would call this a computational microscope that uses hidden Markov models. And I'm not going to go through the math, but we can talk about it afterwards. But here's the idea. This, what you're seeing here is a typical fluorescence trace we get. And you will be hard pressed to tell me when a promoter is on and when the burst ends. But now we can find the most likely trajectory of the promoter throughout the whole time course. Okay. And we can do this for every cell throughout the embryo. Here we're color coding this nuclei according to whether the promoter is on or off. And what I want you to notice is that even in the middle of this stripe, where you have the highest level of expression, you still get bursting. There's no such thing as being on all the time. Okay, so now that we know K on, K off, and R, we can measure it as a function of position, and we can ask, how does the burst frequency vary as you move along the stripe? And what you see is that, sure enough, the burst frequency is modulated in a stripe-like fashion. If you look at the burst duration as a function of position, you see that this is not modulated in any way. But the burst amplitude is modulated also in a stripe-like fashion. So what this tells you is that these 4 transcription factors that are talking to the stripe are somehow talking to some combination of K on and R, but not talking to the K off in any way. And this is more or less consistent with what most people have seen in lots and lots of organisms. Okay. In fact, I would claim that we've gotten pretty good at figuring out which bursting parameters are being regulated. However, I claim that we still have no idea about how this regulation is implemented at the molecular level. What I mean by that is, what happens? How does transcription factor binding trigger and regulate transcriptional bursts? And you can imagine a bunch of different scenarios. These are not mutually exclusive in any way. Where, for example, the transcription factor could have to be there for the whole duration of the burst, or the transcription factor only needs to be there at the beginning of the burst to prime it or something like that. So clearly you need to know something about the binding and its relationship to bursting if you want to make progress towards uncovering the molecular mechanisms of bursting control. So this is hard to do experimentally, though we're making some progress. So first we decided to put on our theorist hat and use theory as a microscope. And the simplest model that I could think of is one where we associate the on state with a state where the activator is found. And with the activator unbinds, then you don't have bursting anymore. So the simple model predicts that the burst frequency is going to be given by the activator binding rate, and the burst duration is going to be given by the activator residence time. So this is a very simple prediction. I know how to measure burst frequency, bursting dynamics. So now I just need to measure binding and unbinding of transcription factors in vivo and compare the 2 timescales. And we did this using an actual very fancy microscope that was built by my colleague Xavier Darzac, this lattice light sheet microscope. And we looked at the binding of bicoid, one of the activators of even skip stride 2. What you see here is a nucleus in the fly here. There's another nucleus, there's a bunch of nuclei here. This little bright spot corresponds to a bicoid molecule being bound, okay? And I don't know if it's going to play that well in the video, but hopefully you can see some flashes of light. Each flash corresponds to a binding event. And what you can do then is ask, how long do these flashes live for? And that's a proxy for the bicoid residence time on the DNA. And what we find is that that's about 1 second, which is a lot faster than the burst duration of 5 minutes that we saw. Okay, so there's a disconnect between the timescales. And this disconnect is not exclusive to binding. Here I'm showing you on the top the dynamics, the timescales measured for bursting in multiple organisms. On the bottom, I'm showing you the timescales of the many biological processes, molecular processes, that you could have blamed for bursting. And what you can see is that most of these processes are faster than the actual bursting dynamics that we observe. So there's some disconnect here. There's something that we're missing. And I'm happy to talk more about the theory in the questions, but there's one complaint that you could raise from the type of testing of the theoretical model we did with lattice light sheet microscopy, which is that when we measure binding, we're measuring binding at a random place in the genome. It could be bicoid could be bound non-specifically, specifically. We don't know what it's doing. But the prediction of our model really has to do with what's happening at the promoter, where you can have multiple binding sites, when you can have cooperativity, and where you could also see what's happening with the rate of transcription at the same time. So great progress has been made on this front in yeast and in mammalian cells, but it's been tricky to do this in flies. Okay, and we have theoretical reasons to believe that at the site of nascent transcript formation, you will get long-lived events, and we can talk about it also in the questions period if anybody's interested. Okay, So how do you actually then go from just measuring binding at a random location to correlating binding and transcriptional output? And so this has led to the development, or to our initial development, of a new microscope that is based on just trying to be a little bit smarter about data analysis, not necessarily getting a better microscope. And to illustrate that, I'm going to switch gears. I'm going to show you a different arrow of the network diagram. The change doesn't really matter, but I just need to tell you some of the names of the molecules, okay? Instead of a strap, we're going to look at a domain of this gene called SNAIL that is expressed along the dorsoventral axis of the embryo. And this gene is activated by the gradient of a transcription factor called dorsal, okay? And if you take data with the MS2 technology, you'll see more or less the same type of dynamics, right? You see the broad domain appearing at the bottom of the embryo, which corresponds to the ventral side of the embryo. So you can try to measure then dorsal venous concentration and SNAIL using MS2, okay? And the first question we ask, based on some really cool fixed tissue data by Ido Golding and Jun Ma, is whether I could see dorsal venous molecules binding to the DNA, to the transcription locus. Basically, would that lead to a higher concentration of dorsal at the site of nascent transcript formation? If you look at a snapshot, the answer is no. This is dorsal concentration throughout the center at the site of nascent transcript formation, and you don't see it. No matter where you look, you don't see anything, okay? Now, the cool thing is that once you start averaging over a few frames, a signal emerges. So you can see how, on average, there is indeed more dorsal at the site of nascent transcript formation, at its target site, than anywhere else in the genome. And it's a concentration that is higher by a factor of but it's like 30% or so, which by our estimations, we're talking about a few molecules, maybe 10 or so. So it turns out that there seems to be a dynamical signature to this enrichment, to this local enrichment. Check it out. So this is the dorsal enrichment dynamics at Hunchback, which is a gene that dorsal doesn't talk to. Okay, so in principle, this is our negative control. This is the dorsal enrichment dynamics at SNAIL, at the locus that it targets. And you see repeatedly these little fluctuations that seem to be higher than what we see for Hunchback. Okay, so one of the things we're exploring now is whether these transient fluctuations actually correspond to unbound to binding events, because they last 5 minutes. So this will be very interesting if these are actually the functional binding events that are leading to bursting. And obviously, once you ask this question, you should also ask the question of whether these binding events also correspond to bursting events. Okay, so great. So this is sorry, just got the 2-minute warning. It threw me off. So this is basically where we want to go. We want to be able to measure this enrichment, which we think is a signature of binding, and correlate it with the output transcriptional bursting. And to me, this is an experiment that I really didn't think we could do we would ever be able to do. And now I'm getting very excited, and probably a big part of my lab is going to switch to trying to think about this, because I really think that this type of experiment can reveal the so-called anatomy of a burst. So we can go beyond just saying there's a burst, and how is it regulated? Is a burst amplitude, duration, or frequency, to actually knowing what are the transcription factors doing before, during, and after a burst. You can even ask, well, what are pioneer transcription factors doing? Do they need to be bound there all the time, or just before a burst? And there are even ways to do some really cool stuff where you could imagine measuring the dynamics of histone modifications during, before, and after a burst, basically on spatio-temporal scales that are just not attainable by genome-wide studies. And this is only possible because we're putting together these different microscopes, okay, in quotes, to reveal the in vivo biochemical information that we need to derive governing equations of cellular decision-making. So just to wrap up, I told you that we're trying to endow the network arrows with molecular and quantitative information that allows us to reach a predictive understanding of developmental decision-making. We're hoping that this will lead us at least to be able to predict how different basis elements of the developmental process are put together, how you make gradient steps and stripes. I showed you how you make a stripe. And obviously one big question is it true that I can take everything apart and put it together, take it apart in these little basis set and put it together to actually predict how a full multicellular organism is made? So with that, I need to acknowledge these guys that I already introduced you to at the beginning of the talk, and my lab in general. It's a fantastic group of people to do science with, and these funding sources that give us the freedom to do crazy stuff, and everybody for your attention, and I'll be very happy to take any questions. Thank you very much.
Wonderful talk. I have a question from Arjun Raj. Love the Markov scope. Question, does the inference require a simple two-state promoter model? How would the inference change if one incorporated additional features like post-burst refractory period?
Yeah, so no, it doesn't. In fact, what I didn't tell you is that when we're looking at a little spot, there's actually two spots on top of each other, because that's how the sister chromatids stay with flies. And we're actually fitting to a three-state model. So you can change it to do fancier things. The question, though, that we've been struggling with is, how do you do model selection? Okay, how do you know which model is right? And we tried to do that for different states. And depending on which measure of model selection you wanted to use, BIC, AIC, cross-validation, you got a different answer. So we're still not entirely sure of how to deal with knowing which model is the right one.
Okay, next question is from Jason Hughes, who asks, can your model be used to interrogate the molecular mechanism behind these gene regulatory events, such as DNA looping or transcriptional condensates, et cetera?
Absolutely. I mean, first of all, from the hidden Markov perspective, one thing is that we're starting to explore is you don't just get burst frequency, but you might get a distribution of burst frequencies. And that already gives you an extra dimension onto which to constrain models of gene expression. One thing that I didn't tell you is that dorsal seems to also show up in clusters. And the clusters move around about the nucleus, and they actually seem to be closer, on average, closer to the site of nascent transcript formation than to non-target genes. So it would be interesting to also correlate bursting with the arrival of these clusters.
Okay, Hernan, I want to ask a question of my own. So you talk about burst times that are about 5 minutes, and in principle, this could be shorter than the loading time. If you think of the length of genes and the speed of RNA polymerase, it might take like 10 minutes until it's completely cosmic before the first RNA polymerase falls off. So do you sometimes get these kind of scenarios, and are you thinking of integrating that into the model?
So you mean that the burst might last longer than the actual time that a polymerase lasts on the DNA, or the other way around?
So the other way around, the burst is shorter. So the promoter it closes before the first RNA polymerases reach the end of the gene.
Yeah. So
you're not getting to plateau.
Yeah. So the whole point of the hidden Markov model is that it deconvolves that contribution. That contribution, the memory of the RNA polymerase on the gene, is what was killing us, and that's that's the whole point of what the hidden Markov model is doing. Now, it requires you knowing the elongation time pretty well. So we've been developing some autocorrelation approaches based on some cool work by Antoine Coulon and others to measure that elongation rate. And I can tell you that there are subtleties, like, we're assuming that the moment the transcription is done, the polymerase falls off, but, termination doesn't happen right away. I mean, there are some subtleties here that as we get fancier and fancier with our experiments, we'll have to figure out.
Thanks.
I also had one question of my own. So what is the best way to prove that this increased transcription factor binding that you see by the time averaging is real binding to the correct site? Is it possible to do it in, like, film the cell and then do some sort of biochemical measurement in that very same cell where you catch it in the act of this time averaging localization? Is that feasible?
Like a single cell ChIP-seq?
Yeah.
Yeah, I think that might be tricky. I think the best bet we have is we have controls that I didn't show you, where we show that dorsal doesn't bind to other, you don't see the enrichment signal in non-binding guys, in non-target guys. And also another experiment we're doing is trying to change the number of dorsal binding sites to see whether the signal correlates with, presumably there's something about occupancy here, right? Eventually the sequences matter. I mean, even if you believe in condensates doing something, like, things need to bind to the DNA. So I think that might be the best way to do it. I don't know how to marry this with genome-wide technologies, just because the timescales are completely off. If the thing stays for 5 minutes, but it only shows up every 5 minutes, it's going to get diluted out so much when you average over the whole embryo and over all time points and such. Sure. But I would like, if somebody has an idea, I would love to hear how to do that.
Okay, let's take one last question from John Murray. At the risk of opening a can of worms, could the longer duration dorsal binding event be a signature of some type of phase separation type event?
I don't really know. I don't really know. Though, as I said, we see that not all transcription factors do this, but dorsal lives in little clusters. So it could be that these enrichment events that we see are the dorsal clusters crashing against the transcription site. So we don't see these clusters forming and like fusing, but we haven't been able to image it for too long. But there is one hypothesis that the enrichment events are the result of the transient visit of one of these clusters to the transcription site.